Okay, guys, this is part 2 of our Lipids and Proteins PowerPoint. This time we're going to talk about proteins. And we should note that they have monomers called amino acids, and that these amino acids are connected. They are connected by a type of peptide bond, which is a form of dehydration synthesis. So in other words, if we have amino acid 1 and amino acid 2, and they're bonded together, that's the peptide bond, and is formed with the loss of water. And then the polymer is called a protein, and we sometimes will call that a polypeptide chain. We will get into later in the course the difference between a polypeptide chain and a protein. Amino acids they have the following structure. This is another one of those areas that's very testable. We always start with our central carbon. Carbons have 4 valence electrons. Then we'll have our carboxyl group. And our amino group, this is just a hydrogen. And then there's an R group. And that's sometimes called a side chain. Or radical. I tend to call it an R group, but you will see it in your textbook and in various biological papers as side chain or radical. There are 20 different types of amino acids, and that means there are 20 different types of R groups, because that's the only thing that differs from one amino acid to another. It's the R group. Amino acids all have the structure. The only way that they're different is one R group is different from another. Yes? Yes. Let's talk about protein structure. So the primary structure is a linear group of amino acids. Those are connected by peptide bonds. So you might have this, for example. Where each of the circles is an amino acid, and they're connected by peptide bonds. The primary structure is theoretical. It's not seen in nature because there are other interactions. Remember that our amino acids look like this. And as they bond together, they form their primary structure. The carboxyl group of one amino acid may interact with the amino group of another amino acid. This causes the secondary structure. So the secondary structure is caused by interactions of one amino acid's carboxyl group with another amino acid's amino group. There are two types, and so let's look at those. The first type is the alpha helix here. And then we'll transition, and the second type is what's called a beta pleated sheet. So you should be looking at a picture of that now. Okay, moving on here. Let's fix that a little bit there. Get excited and start moving the paper all over the place. The tertiary structure is different. It's caused by interactions between the R groups and is globular. It looks like a big wad of protein. Types of R group interactions include covalent bonds. Okay, so far so good. Hydrogen bonds. All right, I'm with you. Ionic bonds, sure. Plus something called the hydrophobic effect. And that should have you go, what? Yes, the hydrophobic effect is when So the hydrophobic effect is when hydrophobic R groups huddle together in order to minimize their contact with water. And then there's also a So this is only seen in the amino acid cysteine, and it's when the sulfur in one cysteine bonds with the sulfur in another cysteine. The quaternary structure. This is when two or more tertiary structures interact. And a good example is hemoglobin. It has 4 tertiary structures. Okay, gang, that's all I've got for you right now. Hopefully you're doing wonderful, and hopefully I'll see you guys shortly. Talk to you later.